So we are going to learn about classical genetics today and the laws of inheritance, how genes are inherited from parents to offsprings, and how do we analyze them? How do we understand, gene function through genetics? There are two major concepts here in the today's lecture. One is, of course, the Mendelian genetics, the classical genetics. So Gregor Mendel was the one who postulated these two laws, law of segregation and law of independent assortment. And Mendel was working in 1800, in 50s, 60s, and he published, I think, in 65. His work, for the first ever time, discovered factors which suggested that, genes, they, he did not use the word gene, but he used the word factors. So this was the first ever time somebody suggested that there are factors within living organisms which control the inheritance of traits or characters from one generation to another one. And nearly 50 years later, we learned that these factors are genes. So why my slide is not changing? So yeah, it's changing. On this slide, you can see already we saw these pictures in last lecture as well. These are corn, and you can see the color of the kernels here is different. Within the same species, within the same kind of corn, you have different colors of kernels. Within, carrots, you have different colors. So an observable feature in a living organism, for example, here you have color of this is the black color, or the color of the seeds, or let's say color of flowers. We call them characters, okay? In genetics, we call them characters. And then, of course, we have characters can be further, distinguished. For example, we have white flowers and purple flowers. So these are specific traits now. So white is a specific trait, purple is a specific trait, but the color of flower is a character. So Mendel was actually observing different characters and traits in the pea plant, and that is the topic of today's lecture. The second important thing, as I mentioned, there are two important things. Second important thing is how we rediscovered Mendel, how we learned what Mendel proposed as factors, whether they are controlled by specific factors. How do we know they are genes? And that is the work from T.H. Morgan, who worked on fruit flies, who brought fruit flies in the lab, and he characterized then genes and relationship of genes with chromosomes, and he went on discovering one after another big breakthrough, the sex chromosomes, autosomes, etc. So today we are going to focus mainly on the Mendelian laws and how can we understand this relationship between genes, chromosomes, genes and genetics and genomes, etc. Essentially how genotype to phenotype relationship can be explained. We will learn precisely what genotype is, what phenotype is in today's lecture. So genetics basically deals with the genes. So study of the genes is called genetics. How do we define genes? I think in last two lectures you very clearly understood what gene is. Genes, they are segments of DNA, a specific segment of DNA which encodes a specific information. Okay, and that information is decoded through transcription and translation, etc. So gene is a specific sequence of DNA on chromosome, and genes, they are heritable. So the chromosomes are passed on from parents to offsprings, and which means genes are inherited from parents to offsprings. Here it's shown two fruit flies, the male, female, and what they are passing on to their future generation are actually these chromosomes, okay, through the process of meiosis and subsequent fertilization. All the characters, all the observable features, like you have blue eyes, you have tall height, you are short, shape of your nose, color of your hairs, complexion, etc. All these are actually encoded by different genes. Here is human karyotype. So karyotype means or the complete chromosome set, a pair of chromosome 1, a pair of chromosome 2, a pair of chromosome 3, keep going, and then X and Y chromosome. Okay, so studying genes and their inheritance, they require a very clear understanding of relationship between genes and chromosomes and how these chromosomes, they are inherited, the processes of inheritance of chromosomes, which we call meiotic cell division, etc. Genetics deals with all of them, and genetics is actually an extremely useful discipline or tool using which we can understand the function of a gene. Gene function can be very beautifully described or understood or dissected using the tools of genetics. So as we learned previously that these are the genes which are responsible for continuity of life in a species, preservation of ancestral characters generation after generation. This is due to genes and inheritance of these genes from one generation to the next generation. And that concept is called heredity. And I explained you last time also, cats will always have kittens, humans will always have babies, dogs will always have pups, and etc. However, due to variation in the genome, we do see some deviations from hereditary. And this deviation does not mean you will start having pups in your family or chimps in your family. Deviation or variation in the genome actually helps us become different from one another within the same species. For example, in humans, no two siblings or two members of the same family, they look alike. Even identical twins, they start becoming different, and they have some different features, observable characters which are different. So this deviation is actually due to the variation in the genome. So what controls heredity, or what is the basis of heredity and what causes variation within a species, is actually genes. The answer lies in genes. Variation in genes that is inherited in future progeny, which keeps moving in a population, and we observe different traits within same species. So a character, I told you, is an observable feature, like for example, flower color, and trait is a particular form of a character, for example, white color of flowers or purple or whatever, or uh, red blood and red blood, the citrus or the uh, normal citrus color. So when these features or these traits, they are heritable. Heritable means something which is passed on from parents to the offspring. When something is passed on from parents to offspring, we call that trait as heritable trait. Now, this slide just shows you some of the traits in different species. For example, here we see eye color, complexion, okay, color of fur of within dogs, height, okay. All these traits, they exist in more or less different species, okay. Now, we have to understand, if we want to understand how these traits, how traits or characters are inherited, we have to understand the fundamental unit of inheritance. Fundamental unit of inheritance. And the fundamental unit of inheritance is actually a gene, because it's the gene which controls color of eyes, gene which controls complexion, of skin, of fur, of height, shape of nose, etc. So we have to understand, if we say there is a gene for eye color, then why don't we all have same eye color? Why we have difference? We have difference because we have different forms of genes. We will have eye color gene, which will be translated into a protein. But this gene, this DNA sequence, which encodes this protein, this gene can exist in alternate forms. Alternate forms. What do I mean by alternate form? For example, this is the eye color gene, and this DNA sequence, it goes like this. It encodes black eyes. But another individual containing the same gene, 100% same gene in terms of its location on the chromosome. Position of a gene on a chromosome is called locus, okay? Same gene in my body is giving me brown or black eyes, but in a different individual, same gene on a different position is giving you blue eyes. In another one, it's green eyes. In another one, it's gray eyes, brown eyes, same gene. But when you go deep down and understand the gene, what is difference between black, blue, green, etc., you may find variation, some nucleotides different than the black one, and that gives you this nucleotide change, gives you a different amino acid for this protein here, and this gene product will give you blue color in the eyes. Similarly, a change or a mutation or a polymorphism at a different position, which results in a different amino acid. In this, all the rest of the amino acids are same in the protein. It gives you green eye colors, gray. So alternate forms of the same gene are called allele. A gene, one gene may exist in several forms, and these different forms of same gene are called alleles. We represent different alleles differently. For example, this one we represent B, this one with B prime, this one with B double prime, this one with. Each allele has a specific symbol, but this symbol will be same, B, but it can be capital B or small b or B1 prime or B2 prime or B3 primes. Now you understand why we see variation within the same species. These are human eyes, but blue eyes, brown, there may be black, there may be green, okay? This is what? This is variation within the same species, okay? So this variation within the same species is due to different alleles in the population. And you can now say allelic variation, which means variation in alleles, will be translated into protein variation, and that protein variation will be visible as observable features, as physical characters, which we call phenotype in a species. Okay, so this will be called B or B prime or B prime. This will be called genotype, the genetic makeup of an individual, and its physical appearance, let's say brown or black and gray or green, that is phenotype. How new alleles they develop, how this variation arises in the genome. This can be very dangerous, no? If DNA starts changing itself, you know, this could be a very essential gene, and this amino acid can be very essential. One may die. But the source of variation is indeed mutations. Mutation is change from one allelic form to another. Capital B changed by mutation in certain nucleotides, it is now B prime or a small b, whatever. We will soon see why small b, why capital B. Okay? So mutations or variation in the genome are the source of variation in a species. And variation in a species, it can have genetic as well as epigenetic basis. At 101 level, I don't talk about epigenetics, but I mean, the ones who are passionate about biology or who are passionate about life sciences, they should go and read how fascinating epigenetics is all about. It's such a fascinating phenomenon. Epigenetics deals with, genes. It does not cause any change in the DNA sequence, no mutation, but still it gives you control over the function of the gene. It can switch on and switch off gene, just like classical mutation. Now, we'll talk about this phenomenon later. Now, how do we learn that, genes are, where I want to go? How did we learn that heritable characters are controlled by genes? How did we learn that genes control different traits? Observable physical features are controlled by genes. How do we learn this? Mendel did some groundbreaking work. Mendel was a monk, a priest in a church in Austria. He attended meeting. He was a genius, great scientific mind. He attended meeting of, I think, Botanical Society or natural scientist. And the conversation in the meeting was, how can we, the question was, how can we improve quality of wool in sheep? At times of Mendel, I mean, the two major empires, Spanish and Austro-Hungarian Empire, and there was an economic, it's always, wars are always due to economics. And there was a loss to Austro-Hungarian Empire who discovered that Spanish are producing very high quality wool, and they have taken all the business of wool. So the scientific community was asked to help improve the quality of the wool in sheep so that we can compete Spanish wool. Mendel came back, and Mendel started doing some experiments. And while he was thinking about this question, he thought, how can I improve the quality of the wool in sheep if I don't know what controls the quality of the wool? Which means he came up with an idea that I have to first understand, discover, or learn about the factors which control a trait, which was wool quality. Okay. He selected pea plants as his model system, and what he wanted to do was to understand if, physical features, observable features, which we know today, phenotype, if they are controlled, if they can be understood, if their inheritance from parents to offspring can be understood. He chose pea plants, and you will learn that he made the first right decision. Question is about sheep, but he is picking up plants. Reasons were because he was a botanist, he understood plants, he understood the structure of the pea flower. So pea flower has the female part, which we call the stigma. This is the ovary, and within the ovary, you see these ovules or seed formation. The male part is the anthers, Okay. So anthers are these sacs. If you pick up of any flower you pick up on campus, so they are like this. These are sacs. Within the sacs, you contain powder-like material, and this powder-like material is called pollen grains. There were petals, like it was a simple flower, not a complex flower. And these pollens, they fall on the stigma, travel through this neck of the stigma and fertilize egg cell here. Okay, fertilization of egg cell or ovule with pollen grain leads to development of a seed. Okay, embryonic seed. Now, this was an intelligent selection because what he did, he could see variation within the pea plant. He could see, pea plants are tall, and he also had dwarf pea plants, purple flowers, and he had white flowers too. He had terminal flower and auxiliary flowers, where flowers will be like in the auxiliary parts. He had yellow seeds color, and he had green seed color. He had round shape of the seed, and he had wrinkled shape of. So he could see variation within the pea plants, and he said, I can do experiments in this model system because it does not take long, and in 8 weeks, hardly 8 weeks, I can harvest seeds and go to next generation, and in another 8 weeks I'll go to second generation, then third generation. But what was more important, he could control these experiments by saying, okay, I'll take purple flower plant and a white flower plant. I'll emasculate. Emasculation means you remove the male parts, the pollen, the anthers, simply with a forceps. And now this flower cannot be pollinated, or this process is called pollination. When pollen grains fall on the stigma and the fertilization takes place, this is called pollination. Okay. When pollen grains of same flower fall on stigma of same flower, we call this self-pollination. Mendel said, I can control the crosses. I emasculate, let's say, all the anthers from here. Now, pollen cannot fertilize white flower. I will cross pollination. I will use cross pollination. I'll take pollens from this plant and throw them on the stigma. This is called cross pollination. And then go to the next progeny to see how the seed develop and what kind of flowers they bear, purple or white. Because now I have genetic material. I mean, he didn't know genetic material, but of course he used pollens from here, fertilized or pollinated white flowers. Very interesting experiment. Simple, but very beautiful experiments. You can simply take these paint brushes, you simply touch them, brush ko anthers ke saath, and the pollens will be released with, they come along the brush, and that brush you would simply rub on the top of the stigma and fertilization takes place. But before setting up experiments, what Mendel did, he said, let us first see if we have true breeding lines, true breeding, or pure lines. Pure lines or true breeding means what he did, he said, okay, I take round seed, I sow them in soil, And in the progeny, do I get all round? If I get 100% rounds, it's a pure line or true breeding line tube. The variation in the pea seed shape was wrinkled seed as well. He said, okay, he took wrinkled seed, sowed them in soil, harvested generation after generation. Wrinkled always produced wrinkled. Rounds always produced rounds. So this is phenotype, round phenotype, wrinkled phenotype. Okay. This is called control experiment. This is going to serve as your reference. Whatever experiments you are going to do, you're going to compare, you're going to use this experiment as reference. Now, Mendel said, okay, I have two pure lines and round and wrinkled. He set up a cross, just like this one, just like this, you know, purple and brown. He took round and wrinkled. He took, let's say, pollens from round or vice versa from wrinkled, and he went to the next generation. To his surprise, everything in the first generation, F1 is called filial generation. F is for filial, first filial generation. Everything is round. Wrinkled, it disappeared. Okay. He said, let's take this round, F1 round. Now, take F1 round and I'm going to self-cross. No more the cross-pollination. I have F1 round, and in the progeny of F1 round, what he found was that wrinkled, it reappeared without any loss in the phenotype of wrinkled. It reappeared in the F2 population. Okay. And round was also there, but wrinkled reappeared just like the original pair. Mendel did these experiments with all 7 different traits I explained you here. You know, I think you wrote, yeah. He used tall plants, tall people. So here he had tall and wrinkled. What he did, he took tall pea plants crossed to dwarf, went through F1, self-pollinated, self-crossed F1, went to F2, and he found all F1s were tall. All F1s, 100% were tall. Dwarf disappeared, but in F2, he found tall to dwarf. Small number of dwarf came back. He took purple versus white flowers. You know, all F1s were purple. When he self-crossed, this symbol, a circle, and this one means self-cross. When he self-crossed F1 plant, in the F2, he again found purple and white with a certain number. All 7 traits, they showed same behavior. G, Ali?
Yes, sir. My question is that in F1, why all of the seeds are not wrinkled? Why all of them are round? Because both have same quantity, I think.
Very good question. This is what Mendel is studying. This is what we are trying to learn. Answer will be in few more slides. Just hold on.
Okay, sir.
Good question. Any other question? Okay. So looking at 7 different traits, height, flower color, seed shape, etc. What Mendel discovered that, you know, one trait, for example, here, round is dominant over wrinkled. He coined, for the first time, he coined the term dominant. He said round dominates because wrinkled is completely disappearing from F1. Round is 100% dominant. Complete disappearance of wrinkled means round is dominant. Now, in F2, when wrinkled reappeared, he coined the term recessive. He said, you know, round is dominant over wrinkled because, you know, in F1, wrinkled completely disappeared. But when wrinkled reappeared in F2, he said it's a recessive trait. We'll soon learn about recessive trait. Okay. And he also said there is no blending of two traits. No blending means, you know, there are no half round or half wrinkled. It's either round or it's either wrinkled. Because before Mendel, blending theory of inheritance, that was very popular. That, you know, from parents to offsprings, you know, traits are blended. Observing all 7 different traits, what Mendel discovered that there in F2 population, when wrinkled reappeared, or white reappeared, or dwarfs reappeared in F2, you know, there is a specific number, and he could count, and he said in F2, ratio between round versus wrinkled, it's 3:1, nearly 3:1. It was never a perfect 3:1. It was like 2.96:1 in round versus wrinkled. In purple versus white, it was 3.15:1. He used his intuition, and remember, intuition has a big role in science. Science is never black and white. We always go sometime in gray areas, and we have to use our intuition. Observing 7 different traits, he said, you know, this ratio is 3:1. This was his intent, and this ratio is phenotypic ratio. What is this ratio? Phenotypic ratio, because we are counting observable trait, round, wrinkled. Round, he said, is dominant, and wrinkled is recessive. Now, he did one very intelligent experiment. He did not stop there. He went to the F2 population. He said, let them grow individually. He randomly took, let's say, 100 seeds. Okay. And randomly, rounds and wrinkles, etc. And he said, okay, are these pure? The F2 rounds, are they pure, or there is something else? What he discovered, that there was a ratio out of 100, you know, ratio was 1 pure round, 1 pure wrinkled, and 2 impure rounds. A round which will give you wrinkled and round, it's impure. No, it's not pure. A wrinkled which gives you 100% wrinkled is pure. And a round which gives you 100% round is pure. So what he said, this F2 3:1 phenotypic ratio, 3 were rounds and 1 was wrinkled, round to wrinkled phenotypic ratio. He said underneath 3:1, actually there is 1:2:1 genotypic ratio. He postulated genotypic ratio. Physically, there is no difference between this round, and this round. But at the genotypic level, at the factors they contain. Mendel did not know about genetics. The genes. He said the factors they control, they are different. They contain, they're different. Physically, these rounds are same. Now, what Mendel did, this is the same thing. 3:1 phenotypic ratio. He said these 3 rounds, they can be further subdivided. 1/4 pure and 2/4 will be impure. And 1/4, 3:1 will be always pure, the wrinkled one. Now comes the point where Mendel changed everything, and Mendel left an impact which is alive till today and will always remain alive as long as we study genetics. Mendel said, I use pure round, I use pure wrinkle. I got F1 where wrinkle disappeared. I self-crossed F1 and wrinkled reappeared. And here I have 1:2:1 genotypic ratio, underneath 3:1 phenotypic ratio. How can I solve this puzzle? What is going on? Mendel said that a character is controlled by factor which exist in pairs. Ji Abdul Basir.
सर मेंडल ने स्टार्ट में जो सा राउंड इस्तेमाल किया है तो मे बी वो इम्प्योर हो. हाउ कैन यू से कि इट इज प्योर?
मेंडल ने स्टार्ट में क्या किया है?
सर राउंड सीड जो F0 में उसने यूज किया है.
हाँ जी.
तो मे बी वो इम्प्योर हो. हाउ कैन यू से कि ये प्योर है?
क्योंकि उसने एक्सपेरिमेंट करने से पहले यहाँ पे कंट्रोल एक्सपेरिमेंट किया था. उसने राउंड को जनरेशन आफ्टर जनरेशन ग्रो किया, वो राउंड ही देता था. ठीक है, तो उसने राउंड के सीड अलग.
ओके सर.
ठीक है. Now what Mendel suggested was, he said a character is controlled by specific factors, and factor which controls a specific trait or character exist in pairs. And he said, since I'm using round as trait phenotype, I will use symbols. And he said, since round is dominant, I will use capital letters to represent dominant traits and recessive ones with small letters. Now, since round character is controlled by a factor, which is symbolized by capital R, factor exists in pair. This is what Mendel suggested. But when a parent passes on factors to its progeny, it only contributes 50%, not 100%, which means this parent can either give this one or this one, not both. And this individual can give this factor or this, but not both. So the characters, they are half when they are passed on to progeny. And the progeny will look like this one, because this round has factor small r coming from this parent and capital R from this parent. And since round is dominant, so capital R dominates and small r effect disappears completely. Now, when you will grow F1 rounds are these ones. you have only 50% can go. Look, either capital R or small r. And this parent, either capital R or small r. Now it depends which one fertilizes what. You can draw a table like this, checkerboard. You say this parent is capital R, small r, this parent capital R, small r. Now, if these two fertilize each other, it will be like this. If this fertilizes this one, it will be like this. If this one with this, it goes like this, or this one. So tell me, what will be phenotype of this one? जिसे मैं encircle कर रहा हूँ. जी उसका
Round.
round. What will be phenotype of this one? जी उसका
Wrinkled.
Wrinkled.
Uswa, you have a question or you have raised?
So I want you to answer the question. It will be wrinkled.
What will be phenotype of this one?
Round.
And this one?
Round.
Very good. So, how many rounds we have? 1, 2, 3, and 1 wrinkle. Now you understand 3:1 ratio, phenotypic ratio 3:1. Now you can very easily see 3:1 phenotypic ratio rounds and 1:2:1 genotypic ratio. Genotypic ratio is 1 pure round, 2 impure rounds, and 1 wrinkled. When you have both the alleles, when you have pair of same alleles, it's called homozygous. Homozygous or homozygote. And when you have two different alleles together, you call this heterozygous or heterozygote. So the combination of alleles which you have, that is called your genotype. So from genotype to phenotype, is all about genetics, is all about life sciences, biology, what we do. Now, this is, we call this, when somebody says you have homozygous round, it means you must think about capital R, capital R. If somebody says you have heterozygous round, you should think about this. If somebody says you have a pure round, it means you have homozygous round. If somebody says you have impure round, it means you are talking about heterozygous. Okay? Gee, there was a question. Go on. Who had a question?
Yes, sir. Sir, ये जो रेशियो है 3:1 phenotype की, ये इतनी perfectly conserved कैसे है? और क्या ये हर किस्म की स्पीशीज में इस तरह ही कंजर्व होती है अगर हम क्रॉस ब्रीडिंग करवाते हैं?
या
अगर हम इसको चेंज कर सकते हैं इस रेशियो को, अगर हम जेनेटिकली पॉसिबल हो अगर?
वेरी गुड क्वेश्चन, वेरी गुड क्वेश्चन. अभी हम देख लेते हैं इसका आंसर आ रहा है आगे. So Mendel started having fun, you know, biologist को, साइंटिस्ट को जब रिजल्ट मिलते हैं ना, वो उसकी जिंदगी का सबसे हसीन तरीन दिन होता है, क्योंकि उसने एक ट्रुथ डिस्कवर किया होता है, उसने सच्चाई, सच तलाश कर लेता है और फिर वो सच को परखता है. He tries to evaluate the truth he has discovered, how far it is true. Mendel said, okay, I have F1 round. You remember F1 round? I'm going to cross F1 round with wrinkle, and you know what? I can predict what will be the phenotype. Bilal, Can you answer what will be the phenotype if you cross F1 round with wrinkle? Bilal. Sheikh Zohaib Ali.
Yes, sir.
जी जिसे क्वेश्चन पूछता हूं वो अपना कैमरा ऑन कर दिया. Can you predict the phenotype of this individual which will result from F1 round cross to wrinkle?
Sir, some of them will be round and some will be wrinkled.
You have to tell me how many will be round and how many will be wrinkled. Prediction तो यही होगी ना?
हाँ जी. एक सेंसर क्रॉस बना लेना.
आपने अपना कैमरा नहीं ऑन किया.
Sir, equal होंगे, equal round, equal wrinkled.
Very good. So it will be 50-50. Very good. What he said, okay, I'm going to take F1 yellow. Yellow was dominant, okay, over green. I'm going to cross with green, and I predict it will be 1:1 ratio. 50% green, 50% yellow. And you know what? His predictions were right. It means what he discovered as factors which exist. So factors control characters. Factors exist in pairs, and from one parent to the progeny, only half of the factor is passed on to progeny. And you know what he said? He said when a parent passes on a factor to its progeny, any of the two factors, whether here it is F1 round was this one heterozygous, this one was this. So here two different alleles are there. They have equal probability. They can segregate. Okay. It's not that one has more preference over other. And this law was called law of segregation. This was about monohybrid crosses or monogenic traits. Monohybrid means, what did you do? Mono means one. You are focusing on a single trait, seed chain, single pair of alleles. And in monohybrid cross, when a pair of alleles is passed on from parents to offspring, they segregate with equal probability. And they both have equal chance to be passed on to next generation. Mendel did these experiments, as I told you, with 7 different traits: round versus wrinkled, yellow, green, purple, white, inflated pods versus constricted pods, green pods, yellow pods, axial and terminal, tall and dwarf. F2 generation phenotypes, and he calculated the numbers and total. And what he said, you know, the ratio, F2 ratio is like near 3. So F2 phenotypic ratio, remember this, F2 phenotypic ratio in a monohybrid cross, ये बहुत important है, in a monohybrid cross will be 3:1. And genotypic ratio, F2 genotypic ratio in a monohybrid cross will be 1:2:1. Clear? यहाँ तक कोई question?
Sir, can you please repeat this point?
So F2 phenotypic ratio, I write down here a problem set जो हमने अभी की है. All the F1 will be round. All the F1 will look like this. We self-cross this, okay? The gametes will be this, this, this. And then in the F2, what we find, 3:1, 3 rounds, 1 wrinkled. I said F2 phenotypic ratio. This is phenotypic ratio. Physical appearance की बात है. F2 phenotypic ratio in a monohybrid cross will be 3:1. And F2 genotypic ratio, F2 genotypic ratio in a monohybrid cross will be 1:2:1, which is underlying this phenotypic ratio. Clear? Did I answer your question?
Yes, sir. Thank you.
Okay. So, Gioswa?
Sir, मेरा question था कि can a mutation recessive allele affect the DNA?
Pardon?
Can mutations in recessive allele affect the DNA overall or not?
Very good question. So, what Uswa is asking, how do you know this allele is dominant or recessive, and what is actually happening at the level of DNA? क्योंकि हमें तो अब 21st सेंचुरी में डीएनए का पता चल गया. मैं इंटेंशनली अभी डीएनए साइड पे नहीं था आ रहा. आई वांटेड यू टू लर्न दिस नॉमेनक्लेचर ऑफ डोमिनेंट, रिसेसिव, हाउ टू सिंबलाइज डिफरेंट ट्रेट्स. फॉर एग्जांपल यहाँ पे आप yeah, you have here yellow and here green. तो जो डोमिनेंट अलील होगा आपने उसे कैपिटल वाई सिंबल देना है. डोमिनेंट प्योर लाइन, होमोजाइगस येलो. अगर हेट्रोजाइगस है तो वो उसका मतलब है स्मॉल. तो जो स्मॉल लेटर्स हैं वो आपके रिसेसिव अलील के लिए. नाउ उसवा इज आस्किंग कि डीएनए लेवल पे क्या चाहिए. डीएनए लेवल पे उसवा इन 21st सेंचुरी व्हाट वी डिस्कवर्ड के ये जो पिक्चर थी जिस स्लाइड पे हम आए थे, आफ्टर डीएनए सीक्वेंसिंग nearly 150 years or 100 years after Mendel, we discovered that जीन तो सेम है दोनों में, same chromosome, same gene, everything is same, but the gene sequence which was supposed to make messenger RNA, उसकी प्रोटीन बनती है और फिर राउंड शेप बनती है, this gene had a defect in its promoter sequence. उसका प्रमोटर उड़ गया हुआ था, म्यूटेशन हो गई थी, इसका मैसेंजर आरएनए नहीं बन रहा, इसलिए वो फिनोटाइप ही नहीं बन रहा. नाउ यू अंडरस्टैंड?
Yes, sir. Thank you.
So, but we should appreciate Mendel that 1865 he published. He must have been working from 1850s onward at least, or 1855 or so. Whatever he discovered, it holds true till today. And today we are learning the molecular basis at the DNA level. जैसे अभी पूछ रही थी कि DNA पे कि वो DNA में म्यूटेशन हो गई है, उसका जीन प्रोडक्ट मैसेंजर आरएनए नहीं बनती, प्रोटीन नहीं बनती, एंड देन सीड शेप डिफेक्टिव हो गई. तो ये क्या हो गया, इसे हम स्मॉल लेटर्स के जब सिंबल देते हैं तो ये क्या है बेसिकली, व्हाट इज दिस एंड दिस, दोनों जीन्स हैं.
Alleles.
Allele. Small I is allele of capital R. And capital I is allele of this. Acha. He did these control crosses, experiments, and in all 7, he made a sound conclusion that there is no irreversible blending. Irreversible blending का मतलब है कि अच्छा आपने येलो ग्रीन को क्रॉस किया है तो जनाब वो इंटरमीडिएट आ गया है और वो इररिवर्सिबल है, वो ऊपर से उसने कहा नहीं, ये दोनों ट्रेट्स आर discontinuous, very clear. When Mendel started predicting, he said, okay, I'm going to use, हमने राउंड को कैपिटल आर क्या है यहाँ पे स्फेरिकल राउंड को कैपिटल एस either way, it's your call, whichever symbol you want to use. Okay, the same way cross. Now there's a new word, test cross. What is test cross? When you are going to take F1 heterozygous and cross it to homozygous recessive parent, that is called test cross. So Muneem, can you tell us the test cross between F1 heterozygous purple with a homozygous recessive parent? क्या करोगे आप? You have to set up test cross of F1 heterozygous purple with a homozygous recessive parent. क्या जीनोटाइप्स होंगे? Muneem, are you there? लो जी, मुनीम का इंटरनेट ही महला डाउन हो गया. Irtaza, are you there? Switch on your camera, please. Irtaza, please switch on your camera. इर्तजा साहब भी लगता है सो रहे हैं. मासूम है, are you there?
Yes, sir. Assalamualaikum.
Walaikum salam. Can you switch on your camera?
I have.
You have switched on your camera?
Yes.
Ji, Masoom. So you have to set up a test cross between F1 heterozygous purple with homozygous recessive parent.
So I think it'll be capital S and then small s for the heterozygous.
Purple trait है, flowers का color.
So the genotype would be capital S and then small s. Isn't that?
ठीक है, ये heterozygous हो गया. तो parent purple homozygous recessive parent कौन सा होगा flower color में? When you cross,
it would be white.
White.
And the genotype for that would be small s for both.
Very good, very good. So you have homozygous recessive means. So let's say we take purple, capital P as dominant, pure breeding. F1 had small p will be white. F1 heterozygous will be this, and If you say test cross, it means homozygous recessive parents से cross करेंगे और ये 50-50 आपके पास ratio आएगी. ठीक है? Okay. So let's now move to the next big thing Mendel discovered. What we learned just is, the monohybrid crosses, and he studied 7 different traits. But what intrigued Mendel was to see if I start studying 2 different traits at the same time, do they interfere with one another? The inheritance of one trait interferes with other. For example, we just saw round versus wrinkled, and we saw 3:1 ratio. He did yellow versus green, and F1 was like this. Self cross of F1, he found 3:1 ratio between 3 yellow, 1 green, or 1:2:1 genotypic ratios. He said, let's, study 2 different traits at the same time. And for that, what he said, okay, I'm going to use yellow and wrinkled, green and round. Whenever you set up crosses, what you should do, let's say I'm saying yellow wrinkled cross to green round. Okay. F1 was all F1. What do you expect in F1? What do you expect in F1? Ji Zishan Farid, please switch on your camera. So what will be the phenotype of F1?
Three to one.
Phenotype of F1. I'm not asking ratio. आवाज नहीं आ रही आपकी. जी Beha Zaidi. Switch on your camera, please. आप म्यूट हैं.
Could you repeat the question, please?
You crossed yellow and wrinkled with green round parent. What will be phenotype of F1?
Sir, I think, would it be green round?
It will be?
Green round?
Beha says green round. Why green round?
Because round was the dominant trait for round versus wrinkled, and green was the dominant color, I think.
No,
So that's why it would
yellow
be
was
green.
dominant.
Oh, then it would be yellow round.
Good, very good. Okay. So whenever you write down a cross, first write down phenotypes. These are phenotypes. And only then you fill in the symbols. Pure yellow wrinkled means capital Y, and wrinkled was small r, small r. Green will be small y because it is recessive and round was this. Then you go through gametes because out of these two alleles, only half of the factors will be passed on. Out of these two, only half. But they will go together, which means capital Y with small r, one combination this Y with this r, this Y with this r, this one with this, and this one with this. तो चार possibilities आपके पास हो गई. So this is what Mendel did. The previous information he had was जैसे बेहा भी हमें बतानी थी कि round was dominant and it was wrinkled was recessive, yellow was dominant, green was recessive. And we already know that in monohybrid cross, we used to get 3:1 ratio between dominant and recessive in F2 and 3:1 between yellow and green in F2. But now we have a state in which we have yellow wrinkled, green round, all F1. So इसके gametes क्या बनेंगे? Capital Y small r, this with this, this with this, this Y with this one, and this Y with this one. तो चार gametes बन गए. Similarly, इसके चारों gametes ये ही बनेंगे, small y, capital R. Now, when fertilization takes place, no matter which one fertilize which gamete, ultimate यही genotype बनेगा, and the phenotype will be yellow round. You self cross F1 and you go to F2. And what Mendel discovered in F2, he discovered 315 round yellow, 108 round green, 101 wrinkled yellow, and 32 wrinkled green. The ratio when you calculate among these phenotypes, these ratios, phenotypic ratios were 9:3:3:1. Now, what is this? Which ratio is this one? This is F2. This is based on phenotypes. And this is a dihybrid cross. So your F2 ratio for a dihybrid cross, phenotypic ratio is 9:3:3:1. Phenotypic. Mendel posed a question. He said, I know when I used to do monohybrid cross, I had 3:1 ratios. Are these 3:1 ratios preserved while going through a dihybrid cross? He did a very simple and intelligent analysis. He said, okay, let's add up all the rounds and all the wrinkled. Forget about, for a moment, keep your eyes blind about yellow and green. Add up round and wrinkled. He added up all rounds, 315, 108, 423. Wrinkled, 101, 32, 133. The ratio turned out to be 3:1. Same for yellow and green. And he concluded that when in a dihybrid cross, factors are being passed on to the progeny, factors assort independently. They don't interfere with one another. They assort independent of each other, and the 3:1 ratios remain preserved. So underneath 9:3:3:1 F2 phenotypic ratio, there are two 3:1 phenotypic ratios. Clear? यहाँ तक? यहाँ तक क्लियर है? कोई क्वेश्चन तो नहीं है? जी ज़िशान. Ji Zishan, you can ask question. आवाज नहीं आ रही आपकी.
Well, sir, now.
Zishan, we cannot hear you. Can you switch on your camera, please, Zishan? Zishan, are you there? Okay. So such a checkerboard we call Punnett scale. वो जो gametes मैंने इधर बनाए थे, you can draw them like this. When you have F2, when you self cross F1, F1 was what? Yellow, round. Now you have to make its gametes, and its gametes will be capital Y, capital R, this one, capital Y, small r, this one, small y, capital R, this one, and small y, small r, this one. And क्योंकि self pollination हो रही है ये. Now you can fill in this table capital Y, capital Y, capital I, capital I, capital Y, capital Y, capital I, small I. So you keep going like this. 16 combinations. Now comes the rediscovery of Mendel. If I ask you to set up a test cross of F1 yellow round with test cross of F1 yellow round, what will you do? You have to set up test cross of F1 yellow round. G Zainab Shafiq. Zainab, are you there?
Yes, sir. Yes, sir.
Switch on your camera, please.
Yes, sir.
So you have to set up a test cross of F1 yellow round. Yellow round means it's a dihybrid cross.
Yeah.
What will you do?
Sir, ये सब crossing होगी proper एक पूरा वो बन. अच्छा, okay. So test cross of F1 yellow round.
हाँ जी.
So sir, basic तो dominant वाले होंगे पहले capital Y, small r, and then हम लोग दूसरा वाला जो लेंगे वो सब recessive लेंगे. So small y, and then small r.
ये test cross setup हो गया?
Yeah.
ये
I
आपने
hope I'm
कहा
correct because as far as I remember test crosses में एक हमारा जो होता है dominant होता है और दूसरा recessive होता है.
मैंने आपको कहा है F1 yellow round.
Yeah.
F1 yellow round इसमें कौन सा जो आपने बताया?
Yellow and then round. Okay, so okay, I get it. This should be capital Y, and then capital R.
If I
This
say
is
it is
yellow
still
round.
wrong, then ये ठीक है कि टेस्ट क्रॉस होमोजाइगस रिसेसिव पेरेंट से क्रॉस किया जाता है. ये तो होमोजाइगस रिसेसिव आपने. अगर मैं कहूँ ये गलत है, then F1 yellow round.
Sir, between yellow wrinkled. Then sir, I'd want you to guide me because I don't really get it.
F1 yellow round कौन सा है? F1 was this one, heterozygous.
Sir, I can't see where you. Acha, okay.
So you
Oh,
cross
acha. It's capital R, small r. Okay.
F1.
That is round. Capital R, small r is
Let's
round.
say you had original parents were like this.
Okay.
ठीक है,
Okay.
F1 में
Yes.
हेट्रोजाइगस होगा। तो ये F1 येलो राउंड है, इसको आप
Heterozygous.
होमोजाइगस रिसेस से पेरेंट से क्रॉस करेंगे तो टेस्ट क्रॉस होगा। ठीक है,
Okay. Yes. Got it. Thank you.
वेलकम। अच्छा जी, सो Mendel published his work in 1865 or so, if I remember correctly. And his work did not get any attention. People did not take him seriously. He lived at the same time when giants like Darwin was there. There were other people who were working on evolution and who were talking about, changes in the genome, selection, blah, blah, blah. It was a very active scientific period, but he remained unknown or nobody for next 50 years or so. It was only in 1900, in early 1900, that I think it was 1905, if I remember correctly, when people discovered meiosis. They discovered that there are cell types in which we see pairs of chromosomes, and there are cell types where we see half the number of chromosomes. Here, 4 chromosomes, 1 chromosome, 2, a pair of chromosomes, 3 and 4, a pair of chromosomes. But then from same organism, the reproductive cells, the gametes, they contained half the number of chromosomes. That was discovery of meiosis. When people saw that, the reproductive cells, the gametic cells are containing half the number of cells, they immediately thought about Mendel. They said, wow, that guy was right. He said, sexually reproducing organisms will pass on half the factor. Here in this cartoon, you have, imagine this is the chromosome pair which contain round gene, this wrinkle gene allele, yellow allele, and green allele. Chromosomes, what Mendel said, factors exist in pairs. Genes are existing in pairs. This is what they discovered in 1905. And why genes are existing in pairs? They said the physical entities which we call chromosomes, they actually exist in pairs. So pair of same chromosome is called homologue. Humans contain 46 chromosomes, which I showed you here. Chromosome 1 exists in pairs, and these two are homologues of each other. Chromosome 2, a totally different piece of DNA, totally different set of genes, has its own pair. Chromosome 3 has pair. So these are homologues. Now, what they discovered that in meiotic cells, in gametic cells, chromosomes, if these two are homologues, diploid, let's say this is monohybrid cross. We are just assuming monohybrid. इसमें बाकी भी chromosome होंगे, लेकिन आपको simplicity के लिए हम करें. Meiosis के टाइम पे क्या होता है? डीएनए रेप्लिकेशन होती है सेल साइकिल में, जब सेल एंटर होता है पूरा डीएनए जो इस क्रोमोसोम पे है ये डुप्लीकेट होता है और इट रिमेन्स एसोसिएटेड, ये सेंट्रोमियर कहलाता है, कॉन्स्ट्रिक्टेड पार्ट। तो जो कैपिटल एस एलील है वो डुप्लीकेट हो गया आफ्टर डीएनए रेप्लिकेशन। इसका होमोलॉग क्रोमोसोम also goes through this. In meiosis 1, the two homologs move to opposite poles and the total number of chromosomes they are reduced to half. यहाँ पे 2 क्रोमोसोम्स थे, अब ये हाफ हो गए, एक इसमें आ गया एक इसमें आ गया, होमोलॉग साइड पे चले गए। This is called meiosis 1. Meiosis is in 2 stages, meiosis 1 and meiosis 2. Meiosis 1 में homolog chromosomes they move to opposite poles and they are total number of chromosomes they are reduced to half. अगर humans में 46 है तो इस first reductional division, इसे हम reductional division भी कहते हैं meiosis 1 को। तो chromosome numbers कितने रह जाएंगे? 23. These 23 chromosomes they undergo now simple mitosis like division जिसे meiosis 2 कहते हैं, और जो sister chromatids हैं ये एक ही क्रोमोसोम है और ये जो 2 हैं इन्हें हम सिस्टर क्रोमाटिड्स कहते हैं, ठीक है। एंड दे मूव टू ऑपोजिट पोल्स एंड दैट्स हाउ फ्रॉम वन सेल द एंड प्रोडक्ट इज 4 डॉटर सेल्स, ईच सेल विद हाफ द नंबर ऑफ क्रोमोसोम्स। यहाँ पे 2 क्रोमोसोम थे, अब इनमें हाफ द नंबर ऑफ क्रोमोसोम्स हो गए। दैट्स हाउ फैक्टर्स आर पास्ड ऑन इन हाफ। If we were talking about independent assortment in dihybrid cross, अब dihybrid cross में क्या है, 2 different traits हम follow कर रहे थे। The 2 homologs they align on the metaphase plate. In meiosis 1, what happens? These homologs will go to opposite pole. This one to this. लेकिन ये random है, independent assortment है। This one can end up with this or this one can end up with this in a gamete. Possibilities है ना, वो इसीलिए हम chromosomes gametes यूं बनाते हैं, this one और this one, यूं करके जो gametes बनाते हैं based on what kind of allelic combination we have. And at the end after meiosis 2 you have ये चारों gamete type, capital Y, capital R, small y, capital R, etc. The inheritance of different alleles they assort independently. Metaphase spread पे ये वाला chromosome जब reductional division होती है, capital S may go with small y वाले chromosome के साथ, or it is also possible capital S वाला ends up together with capital Y. तो ये independent assortment. So what is time now? अब्दुल्ला कितने बजे तक क्लास होती है?
ये 5:30 तक होगी.
Yeah, so we still have 13 minutes. Now, what Mendel discovered that, whatever Mendel discovered, it was proven by discovery of meiosis, and we could see that indeed, the chromosomes, they are reduced to half during meiosis, and these chromosomes, they can shuffle or assort independently in the traits or characters, blue eyes, or whatever height of pea plants, whatever Mendel discovered, it was 100% truth. However, the next question, as I say, in science, every question brings new answer to every question brings or raises a new question. Now, people posed a question that what if round and yellow, they reside on the same chromosome? if round and yellow, they are on two independent chromosomes. So these are two homologs, okay? Homolog of round and wrinkled carrying alleles, or green and yellow one. So if we have, I try to draw it here, let's say the original parents, yeah, this one we can write here. Here we can write. We started capital Y, and wrinkled. This was yellow wrinkled parent cross to small y, and a different chromosome round. This was green round. In F1, yellow round will look like this. इधर से small y आ जाएगा और दूसरा independent chromosome आ जाएगा capital R, small r. Clear? And you can make how the F2 will be. Now the question is, they say if we have yellow and a different chromosome, wrinkled and round, you know, law of independent assortment will be true because we have 4 different kinds of gametes. Gametes kaun se the? Yellow. Original parents, not F1. Original parents ke kaun se the? Yellow wrinkled allele. Then green round. और F2 में हमें क्या मिले थे? अगर हम F2 में चले जाएं तो हमें नॉन पेरेंटल्स B मिले थे, which was wrinkled yellow and wrinkled green. ठीक है? Which means wrinkled green is like this, and fourth one will be what? Capital Y small r, capital Y small r, capital R small y, and it should be this one. Now these two make sense because they were in the original parents and we call them parentals, parental gametes. These ones are non-parental, they were not in parentals. We call them non-parentals because they are result of F1 self-cross and independent assortment because we have capital Y with, यहाँ पे तो gamete यूं होगा ना, sorry, capital Y go in one gamete with capital R, capital Y go with small r. This was original parental. This is now non-parental gamete. Small y going with small r. This is non-parental, and small y with capital R is a parental. This is true. These gametes will be 25% each, or 1/4. 25% each या 1/4 एक ही बात होगी. But the question they raised was, what if yellow and round wrinkled alleles are on the same chromosome, So according to Mendel, After you go through such a cross, now we write down, for example, yellow. Yellow wrinkled क्या होगा? Pure breed yellow. Wrinkled will be smaller. Cross to green and rounds. Gametes F1 में जब बनेगा तो F1 में क्या आ गया? Yellow. ये क्रोमोसोम आ गया एंड ये वाला है. इसको जब आप क्रॉस करोगे, सेल्फ क्रॉस करोगे, तो F2 में आपके पास क्या आएगा? आपके पास 50-50 आएंगे पेरेंटल अकॉर्डिंग टू मेंडल. 25% ईच या 1/4 ईच नहीं आएगा, ये हाफ ईच हो जाएगा पेरेंटल. एंड नॉन पेरेंटल कोई भी नहीं होंगे अकॉर्डिंग टू मेंडल. So answers to these questions were found in Morgan's lab, and Morgan was the one, T.H. Morgan, Thomas Hunt Morgan. He was the one who dared to find and answer these difficult questions. He brought in fruit flies into labs. This is Morgan's lab. And आप कभी bio lab में आओगे तो you will see we grow so many tons of flies. Why we grow flies? Because flies are one of the beautiful model systems. Mendel used pea plants. It requires 8 weeks, 8 to 12 weeks. Flies require just 10 days from one generation to the next. And when you are doing genetic analysis, I think flies are one of the beautiful systems. So I urge you all that you go back today and go through this crossing scheme. I quickly go through and I tell you what Mendel did. Mendel had a mutant fly whose body color, this is black and vestigial. Vestigial is a phenotype whose wings were almost gone and body color was abnormally black. And this is the normal wild type. Wild type हम कहते हैं flies या wild type कोई भी organism जो कि normal predominant phenotype होता है, जैसे round seeds in pea plants. This was a mutant. He crossed them and he was expecting, he knew that brown body color, so he knew that black is vestigial, black is homozygous recessive trait, and vestigial is also a recessive trait. Monohybrid crosses usne set up kiye. But in F2 when he went, he discovered non-parentals, what he called recombinants. We will talk about them in detail in the next lecture. Okay? Now, what Mendel, according to Mendel, if vestigial and black are on the same chromosome, Morgan should have had discovered only parentals, which means, 50% black and 50% vestigial. Okay. But, 50% wild type and 50% black vestigial he should have received if both black and vestigial are on the same chromosome. But he did not discover, he did not find, 25% each. There was a big skewed ratio. Recombinants or non-parentals were 1:1, and here was also 1:1, but it was not 1:1:1:1. The non-parentals were 50% each. 1:1 ratio. Parentals were 1:1, but there was a big difference between parentals and non-parentals. What he did, he took wild type flies and homozygous black vestigial recessive. Plus means this is your call. if you gave black body color, small b, recessive trait, this can be capital B, or if you just write down plus, it's also okay. Or B plus, and on this side, if you write capital B, but with minus, that will also be okay. But for ease, I recommend go with capital B, small b. Here, small vg is vestigial. This wings are gone. And here it will be capital VG. We don't know they are on the same chromosome or no. Okay. You went to next generation, F1, and everything was normal. Capital B is dominant. From monohybrid crosses, he knew brown body color is dominant over black, and normal wings are dominant over vestigial. F1 population was test crossed. Test cross is very useful for genetics because you are fully confident about the genotype of or genetic makeup of that homozygous recessive parent. What it did, he said, okay, capital P is a यूं कर लेते. The parentals, what are the parentals? Capital B VG is one parental and the other was small b VG. According to Mendel, he should have received these 50-50. And if they reside on same chromosome, then 50-50. If they reside on different chromosome, they should be 25% each. Law of independent assortment in a dihybrid cross. But when they counted, what he discovered, 965, 944 parentals, 206, 185 the other parentals. Ratio between non-parentals was 1:1. Ratio between parentals was 1:1. What he did, he said, okay, let's count the ratio, frequency or percentage. Non-parentals, 206 plus 105 divided by total number of flies. Total number kya hoga? Parentals plus non-parentals. He discovered it's 17%. What he did next was, he set up a different cross. And setting up a different cross means previously one of the parent was completely wild type. What he did, he brought vestigial in one parent and wild type for body color. Black and then wild type for vestigial. Again, he went through F1. In F2, he found again the same numbers, and the ratio of non-parentals here was also 17%. Mendel said they are definitely not residing on different chromosomes. Had they been lying on different chromosomes, they should have assorted independently, and we should have discovered, 25% each these phenotypes. Since we are getting a smaller number of non-parentals, they are most likely on the same chromosome. And this puzzle, this enigma was resolved or solved by one of the sophomore students. Sophomore student, I'm repeating. Many postdocs, PhDs, they ran away solving this problem. What sophomore said, his name was Stratavant. What Stratavant said, he said, both the capital B, they are on the same. So both the genes for black and the vestigial, they are lying on the same chromosome. At meiosis, when the homologs, they come together close to each other, they undergo recombination. They physically exchange their DNA segment. And as a result, what happens, after DNA replication, before the alignment on metaphase, they make a crisscross. The homologs, I'm saying, only the homolog chromosomes like this. So now here you have capital B, and here you have VG. The result of this recombination will be capital B, small VG. The one which do not undergo recombination, it will remain like this. Parental. This is parental. This is non-parental now. A reciprocal of this crossover will be this one with this one. Here we have small b, and here we have capital VG. The result is small b, capital VG. This is also non-parental, and this was parental. He said due to this crossing over, this phenomenon of crossing over during meiosis between homolog chromosomes, we get recombinants, and he called them result of genetic recombination. This was a new discovery that genes which reside on the same chromosome, they can undergo recombination. This was a deviation from independent assortment, because independent assortment concluded that whenever you will deal with two different traits, the factors which control the two different traits, they assort independently. But here we are dealing with two different traits. The factors are not assorting independently. We are not finding 25% each or 1/4 each, but we are finding, non-parentals in a specific ratio, and that is due to the genetic recombination. So we end up here at this stage, and tomorrow we'll pick up how do we prove genes they indeed reside on chromosomes. Any question? Any question?
Yes, sir.
G.
Sir, why do we have 3 alleles for our blood group?
Why we have?
3 alleles for our blood group.
Very good question. Evolutionary, it's very useful. And that's, if you know about blood groups do not behave in completely dominant and recessive manner. That's a different phenomenon. We call this codominance. If A and B come together, the blood group becomes AB. Okay?
Okay, sir.
We don't have complete dominance के A जो है वो dominate कर जाएगा, क्योंकि B allele की भी protein या antigen express हो रही होती है, and that may be lethal. तो वो evolutionary जो selection है, या मैं तो उसे अल्लाह कहता हूँ जिसने ये पूरी engineering की हुई है, बड़ी ही intelligent है, इतनी amazing है कि at the end you just say Subhan Allah. वो codominance है, that is not completely dominant trait.
Okay, sir. Thank you so much.
Any other question? So if there are no more questions, I wish you all a wonderful evening, and I am uploading all the lectures on the same playlist. Allah Hafiz.